Okay, it's about 1 minute past the hour, so I think we will get started. Good afternoon and good evening, everybody. My name is Sarah DeFeo, and I am the Vice President of Scientific Affairs and Programs here at Ovarian Cancer Research Fund Alliance. And I want to thank all of you for joining us today as we continue our webinar series for 2017 with today's presentation by 3 OCRFA-funded scientists. OCRFA's webinar series has been supported by Genentech, so a big thank you to Genentech for making this and all of our webinars possible. So just a little about our organization. Ovarian Cancer Research Fund Alliance is the largest global organization dedicated to fighting ovarian cancer. We advance research to prevent, treat, and defeat ovarian cancer, support women and their families before, during, and beyond diagnosis, and work with all levels of government to ensure ovarian cancer is a priority. So before we get started, I have just a few housekeeping notes to go through. All listeners will be in a listen-only mode throughout the presentation, all 3 presentations. We will have some time for questions after the 3 presentations are over. So if you think of a question as you're listening, you can type it into the Q&A box, which you will see on the right-hand side of your screen. I'll see the questions, and we'll select some to ask our presenters at the end of the presentations. You don't need to wait until the presentations are over to submit your questions. You can just type them in as we go along and as you think of them. And also, this webinar is being recorded as well as the slides, so it will all be available on the OCRFA website starting tomorrow. So if you missed something, don't worry. You can always go back and listen to it again. So now, about our presenters. We're lucky to have 3 wonderful ovarian cancer researchers joining us today. Dr. Ron Chandler from Michigan State University, Dr. Wei Shen from the Van Andel Research Institute, which is also up in Michigan, and Dr. Michael Goldberg from Dana-Farber Cancer Institute. They'll each talk to us for about 10 to 15 minutes and tell us about what they're working on and the role that their project plays in advancing ovarian cancer research. So each of the scientists we'll be hearing from today is the recipient of Ovarian Cancer Research Fund Alliance's Liz Tilberis Early Career Award. This is one of the grants that we give. It's a prestigious grant. It's for researchers who are, as the name of the award would suggest, early in their careers. And while they're young, they are what we consider, quote, independent investigators, meaning that they are running their research labs of their own. They're pursuing their own ideas. They have a team of more junior researchers, usually postdocs, postdoctoral fellows, who are working with them. And the folks we're going to hear from tonight, like many of the researchers we fund, are doing what we call basic science research. Now, this is not research that is done in people, like a clinical trial is. Basic science research takes place in a laboratory, and by generating information and insights, it provides a foundation of knowledge for all the other science, including clinical research, that follows. Basic science research is critical because all major advances that help patients, like, say, new cancer treatments, are built on the foundation of basic science and in the research laboratories like the ones run by our speakers this evening. At OCRFA, we believe that advancing this research and supporting the scientists that do it is really essential. And while we've made great strides in the fight against ovarian cancer, we still have a lot to learn. And our hope here at OCRFA is that the research that we fund today will help fuel the discoveries of tomorrow. And Drs. Chandler, Wei, and Goldberg are 3 examples of the next generation of researchers who are really leading that charge. And we are very proud to support their work. So let's get down to it. We're going to start with Dr. Chandler tonight. I'm going to keep the speakers' bios very short. Dr. Ronald Chandler is an assistant professor in the Department of Obstetrics, Gynecology, and Reproductive Biology at Michigan State University. He received his PhD in molecular physiology and biophysics from Vanderbilt University and did his postdoctoral work at the University of North Carolina. So Dr. Chandler, I am going to pass the mic to you, and you can start your presentation when you're ready.
Can everybody hear me?
Yep, we can hear you, and I can see your slides. Great.
Great. So first I want to say thanks to Sarah and also the Ovarian Cancer Research Fund Alliance for their funding for my research, but also their support and advocacy for ovarian cancer patients. So today I'm going to tell you about my ovarian cancer research story. Can everybody see the pointer?
Yes, we can see it.
Great. So there's been several milestones in ovarian cancer treatment over the past 4 decades. Some of you may be familiar with some of these landmark discoveries. These discoveries have led to an overall improvement in our 5-year survival rate, but we still have a long ways to go with this. So currently, roughly about 22,000 women will be diagnosed with ovarian cancer this year. The 5-year survival rate still hovers around 15 or 50%. So 2 in 4 women live at least 5 years after their diagnosis. So I like to think that every cloud has a silver lining, and I think the silver lining for ovarian cancer is going to be technology. The technology that I'm going to talk to you about today, just briefly, is called NextGen sequencing technology. So what this technology has allowed us to do is kind of peel away the kind of molecular layers of ovarian cancer and identify unique features that may be associated with the disease. This includes gene mutations. So a lot of these quote-unquote omics characterizations have been performed as part of a government-funded cancer genome atlas. So Dr. Wei Shin is going to talk to you about this in the next presentation. So the next-gen sequencing technology and a lot of these omics characterizations have been applied to numerous cancers, including ovarian cancer, and roughly about 12,000 tumors have been sequenced to date. So you may be asking how this next-gen sequencing technology may impact cancer treatment, and in particular ovarian cancer treatment. So this is often referred to as the Precision Medicine Initiative or the Personalized Medicine Initiative. So just shown as kind of a cartoon of what precision medicine really looks like. So you can imagine that a patient comes into the clinic, and if the doctor agrees to have their tumor sequenced, they may identify a mutation. And let's say in ovarian cancer, they identify a BRCA1 mutation. Well, that mutation in that tumor may provide the doctor with the information he or she needs to treat that patient. In the case of a BRCA1 mutation, you may treat that ovarian cancer patient with a PARP inhibitor because we know PARP inhibitors work really well on cancers that have these BRCA1 mutations. And the same may be true for other tumor mutations that they may be finding. This allows us to tailor our treatment really to the patient based on the molecular features that are associated with the disease. So one thing that we've identified throughout a lot of these next-gen sequencing technology and these approaches that we've been utilizing is that there are classes of genes called tumor suppressors that can become mutated in cancer. So we see a lot of these tumor suppressor genes in these next-gen sequencing studies that are mutated. So normally these genes prevent cancer, so you can imagine them being the kind of the brakes on this car. However, what happens in cancer is you often acquire your first mutation. This may mutate one of your alleles or one of your chromosome pairs, which may make you a susceptible carrier. And just to provide some analogy, you may be, in this case, a BRCA1 carrier or what have you. But in the case of cancer, you acquire a second mutation or a second hit, and you lose both functional copies of these tumor suppressor genes. And this releases this breaking power of the tumor suppressor, and then you can develop cancer. So just shown here the ranking of the top 20 mutated tumor suppressor genes in the Cancer Genome Atlas. So this is, basically identified through all these next-gen sequencing technologies or next-gen sequencing studies. It doesn't matter what these genes are. I just want to point out that there are classes of genes that were previously unknown to biologists that can function as tumor suppressors, which are shown here in red. So all of these genes shown here in red regulate this molecule shown here. So this is called a nucleosome. So nucleosomes consist of DNA, which is shown here in gray on the periphery, wrapped around a histone octamer core, or essentially 8 histone proteins in the center here. So one way that I like to depict the nucleosome is by basically thinking about a spool of thread or a spool of wire. So nucleosomes are the basic unit of DNA packaging, and what these protein DNA interactions allow us to do is to package our DNA, all of our DNA, into a small size within the cell. So these provide kind of the packaging for our DNA. So these nucleosome spools, if you will, are organized throughout the genome. They can form higher ordered chromatin structures, which ultimately leads to the formation of our chromosomes. And an easy way to visualize this is to think of beads on a string. So this was depicted very famously by Owens and Owens in 1974, and just shown here is an electron micrograph of nucleosomes peppered throughout these strings of DNA, which are shown here. So what the TCJ and all this next-gen sequencing is telling us is that to prevent cancer, we must properly package our DNA in the cell. And another way to think about this is to think about an old VHS tape. We all know them, we all used them when we were children, or maybe as adults, but these VHS tapes allow us to package all of this magnetic tape, which would be analogous to DNA, if you will, into this cassette so it can be read properly. And what happens in cancer is all of this DNA and all this nucleosome packaging becomes disorganized and essentially really runs amok on the cell and leads to a condition where this DNA may or may not be read properly. So cancer is really defined by a disorganized chromatin structure and nucleosome structure. So one of the questions that my lab is trying to address is, does disruption of genes involved in nucleosome regulation of DNA packaging actually cause cancer? So the gene we like to study is called ARID1A, which is a top tumor suppressor gene that was identified through these next-gen sequencing technologies and studies that were performed. So ARID1A is unique amongst other tumor suppressors in that it's mutated at a very high rate in ovarian cancers that are often associated with endometriosis. These are often referred to as the clear cell and endometrioid subtypes. So both of these subtypes are often associated with endometriosis, which causes pain, inflammation, infertility, but is considered a benign disease in itself. And we're trying to understand why a subset of women with endometriosis go on to develop some of the most severe cases of ovarian cancer in the lab. So one of the approaches that we like to use in the lab is we like to use genetic mouse models. What we find is that if we induce ARID1A mutations in mouse ovaries, we can essentially cause this mouse to develop ovarian cancer. And just shown here is an image, a gross image of a mouse with an ovarian tumor. So we specifically place these ARID1A mutations in this ovary shown here. You can see a large tumor growing. The contralateral or control ovary is shown here by this asterisk. So these animals can go on to develop ovarian tumors, but importantly, they also manifest a lot of the clinical features that are often associated with ovarian cancer, which includes the formation of ascites or fluid buildup in the peritoneal space. In this case, this mouse has hemorrhagic ascites or ascites that essentially is blood tinged or has blood in it. So some of the ways we utilize these mouse models is shown here schematically, and I'm not going to go into all the details, but the beauty of using these genetic mouse models is we can really understand the early stages of cancer, or those stages of initiation that often occur. In the clinic, a lot of times when a patient comes in, they have had ovarian cancer for a while. We can't go back in time to understand the earliest stages of the disease. So we use these mouse models to do just that and look at cancer initiation. Because these are genetic mouse models, we can control for the genetics in these mice. We can also use tumor cells from these mice in drug screens to look for maybe the potential for novel drug targets in the disease. And most importantly, since a lot of these mouse models mimic the pathology of the disease throughout its entire history of progression, and we can also use these models to understand mechanisms of metastasis. Ultimately, this is what we want to understand in ovarian cancer, is how these cells spread throughout the peritoneal space. And importantly, another point of consideration is that one of the big pushes in the ovarian cancer field is ovarian histotype-specific treatment. So we know that ARID1A is mutated in clear cell and endometrioid ovarian subtypes. I'm really interested in understanding or using the mouse models to explore histotype-specific treatment rationale in these patients. So finally, this is my last slide, but I want to just kind of leave it with some current challenges in ovarian cancer. Just shown here in 3 bullet points. The first one is we obviously need to improve risk assessment detection methods. That's the first and foremost. We need to detect this disease very early on. in the body. Along those same lines, we need a better understanding of the cellular origins of the disease. As many of you may already know, ovarian cancer is not thought to arise from the ovaries itself and may come from other regions within the female reproductive tract. So it's really important to understand the origins of the disease as well. And finally, probably the most important aspect of the research we're doing is that we're trying to utilize mouse models to better understand and implement new treatment paradigms to combat chemoresistance and metastatic spread. Ultimately, this is what we want to do. We want to basically identify those patients or gene mutations that may lead to chemoresistance or maybe promote metastatic spread. So with that, I'd like to thank all of the attendees for staying with us tonight and allowing me to present my work. And I'd like to thank you for your attention and support. And thanks to Sarah for organizing the webinar today. Thank you.
Thank you, Dr. Chandler. So again, we're going to hold off on all questions until the end. But if you have a question, feel free to type it in now. So next up, we'll hear from Dr. Hui Shen. Dr. Shen joined the Van Andel Research Institute Center for Epigenetics as an assistant professor in 2014. Prior to her move to Lansing, Michigan, she completed her graduate studies at the University of Southern California, where she received training in both cancer biology and biostatistics. And her undergraduate degree is from Nanjing University in China. So, Dr. Shen, I'm going to make you the presenter, and you can get started.
Well, thank you so much, Sarah, for the introduction. Can you hear me well?
Yes, you're a little quiet, but I can hear you.
Okay, I will try to speak a little bit loudly then. I usually don't speak with high volume, so that will be a challenge for me, but I will try. So thank you, everyone, for taking the time to call in and listen to the three of us sharing our work. And it's really a great honor for me to be sharing a little bit about of my research with you tonight. And so the title that I give to my presentation tonight is called Charting the Dark Matter in Ovarian Cancer. And I will explain in a little bit what the dark matter is. And the two keywords of my research are epigenetics and bioinformatics. Probably neither are words that you deal with on a daily basis, but I will try to explain them as well as I can. Epigenetics is really hot now, and this cartoon is funny, but I actually see it quite often if I go to a meeting and one of the audience asks the presenter what they thought something might be, what the reason for that something might be, and the researcher will sometimes they say, well, I don't know, but maybe it's epigenetics. So it is really hot topic now, both in research and also in popular media. So as you can see on this slide, on the cover of Time magazine, I think it was from 2010, the title is Why Your DNA Isn't Your Destiny, and they were talking about this new science of epigenetics. It's really attracting quite some attention in the popular media as well. But unfortunately, actually, there's a misuse and misunderstanding of this terminology in the popular media. So I'm going to talk a little bit about what actually epigenetic is, at least what we use it in the scientific research community. So let's start with genes and genetics. So in our bodies, we have 40 trillion cells. And within each cell, we have 23 pairs of chromosomes. And as we saw earlier, that we receive one set from our dad and one set from our moms. And in each set of these chromosomes, we have about 30,000 genes. And that's really how you define genes. And the numbers could actually be bigger based on how you actually define a gene. And so each set of these chromosomes consists of about 15 million nucleosomes. And Ron just talked a little bit about what a nucleosome is. It's DNA wrapped around a protein histone core. So that's the structure called nucleosome. And if you just look at the DNA itself, in each cell, we have 2 times 3 feet of DNA in that cell. And that is, if you look at the base pairs that encode the genetic sequence, we actually have 2 times 3 billion base pairs in each cell. And this complete genetic code from a cell is called a genome. And to put it in perspective, if we add up all the DNA in our body, it's approximately 45 billion miles of DNA, and that's 250 times back and forth from the sun and about 50,000 times to the moon and back. So that's a lot of genetic information stored in our cells. But if you think about it, we all start with from one cell, that is the fertilized egg, and we all have the same set of, so that means that all of our cells actually inherit the same set of genetic code. But when we think about a mature human body, there are different cell types in the body that includes neurons and keratinocytes. And these cells have exactly the same DNA code, but they're very different. And how is that possible? And how do you make different cell types with the same set of genes? Well, the answer is that you turn on and off different sets of genes in different cell types. And the way to do it is through epigenetic marks. So those are modifications that are added to our DNA to turn on and off genes. And the epigenetic mark that I study is called DNA methylation. By analogy, genetic is like the hardware, which contains all the information, and epigenetics is like the software that instructs the cell how to use the genetic code. So that's epigenetics that I'm trying to explain, and I hope that's clear. And why is epigenetics important in cancer? When you think about these genes that are called tumor suppressors, so your cells need the tumor suppressor genes to be functional in order to keep the cells in check. So Ron talked about BRCA1, and that's a famous tumor suppressor gene for ovarian cancer. And in the cell of origin, the genes are turned on and off like they're programmed. So everything is functioning normally in an order. But in cancer, what happens is that you can have a genetic mutation that changes the genetic sequence and that alters the gene function, or you can have an epigenetic silencing event that turns off the tumor suppressor gene. So epigenetic silencing is seen more as an alternative to the more well-known genetic mutation in tumorigenesis. So when you look at all the genes, earlier I was talking about how many genes there are in the cell. So if you look at all of them together, so we move from single gene epigenetics to epigenome, and we use microarray chips or sequencer to generate all the data. And that's a lot of data that's generated. If you earlier I talked about how many base pairs there are in the genome. So if we just print these letters out, it will take a century to read, basically. So this is where bioinformatics comes in. So that's the other keywords of my research. So I use computers to mine all these data that are generated with the high throughput technologies and try to find genes that are important in the development of a disease. And this slide, Ron said earlier that I'll be talking about the Cancer Genome Atlas. I'm sorry, Ron, sorry to let you down. I was involved in this consortium, but today, because of the time constraint, I was actually not going to introduce this consortium. But this result is actually from our work as part of the Cancer Genome Atlas project. And what you see is that, so this is the one gene, BRCA1, that came out of the genome screening of epigenetic silencing events with the microarray technology that was used back then. So what we saw is that this important tumor suppressor is turned off by an epigenetic mechanism quite often, so in a substantial subset of the tumors. If you look at the top left plot, it's the expression level of this gene versus the promoter methylation level, the epigenetic mark that I study, that's the x-axis, and each dot is one tumor. And what you can see is that there's this group of tumors here. They're colored blue. They have high methylation and low expression. And there are blue and purple dots, actually green and purple dots that represent genetic mutation events, so including both germline and somatic mutation events. And you can see that they're mutually exclusive with epigenetic silencing events. And if you look at how much, what's the fraction of these ovarian cancer cases that they each explain, BRCA1 promoter methylation explains 11% of all the cases, and BRCA1 mutation and BRCA2 mutation, they also explain 11%. So epigenetic silencing is actually at least as important as the mutation events. When we did this genomic screening for different tumor suppressor genes for different cancer types, here, you don't have to read all the labels and everything. I just want to show you that so there are 3 genes highlighted here, but we actually did it for all of the genes and all the tumor suppressor genes that we identified. And this is a bar chart that summarizes the mechanism to inactivate these important tumor suppressor genes either by epigenetics, which is red, or genetic events like deletion and mutation. Mutation is shown in blue. And what you can see is that the BRCA1 gene is inactivated in 2 particular cancer types or cancer subtypes throughout all the cancer types that we studied. That's the basal-like or triple-negative breast cancer and the serous subtype of ovarian cancer. They share a lot of similarities, and you can see how important BRCA1, both genetic and epigenetic events, are important for these 2 types of cancers. Recently, we did, through a collaboration with the Broad Institute, we showed that the BRCA1 epigenetic silencing have the same mutation signature as the BRCA1 mutations and BRCA2 mutations. So this group is the tumors with BRCA1 epigenetic silencing, and on the very left we have the tumors that don't have any BRCA alteration, and you can see that no matter it's genetic or epigenetic mechanisms that inactivate genes in this pathway, you get the same elevation of what they call signature 3. And this is a possible indicator of response to PARP inhibitors. People have been focusing on BRCA1 and 2 mutations, but this shows that epigenetic mechanism is as important. So why studying the ovarian cancer epigenome? So it tells us how does the cancer arise by identifying these driver epigenetic alterations, and it could shed light on how should the cancer be treated. We may be able to find druggable targets or predictive markers like promoter BRCA1 silencing, and it can probably tell us where the cancer comes from. So to solve the self-origin debate for ovarian cancer, Ron showed self-origin as one of the current challenges for ovarian cancer research. And I showed earlier how epigenetics is what controls the cell identity, so that the epigenetics in the tumor register where they come from as well. And this information could also be used in other ways, like early detection of the disease. My research goal basically includes developing bioinformatics tools that can be used by the entire research community, and to generate full maps for epigenetic alterations in these different histological subtypes, and as well as the potential cell of origin for ovarian cancer, to understand what contributes to the development of each subtype of ovarian cancer, and use these maps to help with prevention, early detection and intervention, disease subtyping, and what Ron talked about, personalized medicine and treatment, prognosis, and eventually generate clinically applicable markers. Well, I would like to really thank OCRFA for the funding, because as a junior faculty, it's really, it's not as easy as the more established investigator to get funding from conventional resources like the government. And this award that we receive from OCRFA is really a big help to get our research started. And I'm really grateful for OCRFA to give me that award. And I would like to thank patients and survivors for ovarian cancer, and all the clinical samples that we study were obtained through generous donations from patients with consent. And I would also like to thank all the ovarian cancer advocates and everyone that cares about and helps in the fight against ovarian cancer. And really, nothing that we as researchers do in this battle could be done without you. Thank you very much.
Thank you very much, Dr. Wei. Again, I see some of you are asking questions. That's great. You can just type them in. So last but definitely not least, we have Dr. Michael Goldberg. Dr. Goldberg is an assistant professor in the Department of Cancer Immunology and Virology at Dana-Farber Cancer Institute. His undergraduate degree is in biological chemistry from the University of Toronto, and he subsequently received an MPhil in bioscience enterprise from the University of Cambridge. And finally, he completed his PhD in biological chemistry at the Massachusetts Institute of Technology. So Dr. Goldberg, I am going to pass the mic to you, make you the presenter, and you can get started. I'm going to unmute you as well. There you go.
Thank you so much. So can you see my screen as full screen, Sarah? Or
not as full screen. We see it in just sort of regular. Yep, there you go.
Is that better?
It is. We can see the next slide ahead, but that's fine. Looks fine. It's a good-looking family.
Thanks. That doesn't help. I have kind of two screens here, and I think you guys are just seeing.
I think we're seeing both, but that's okay. Actually, we can see the slides just fine. We can just see one. We can just see two at a time.
I'm really sorry about that. I don't know how to fix that.
That's okay.
But in any case, I want to thank you, Sarah, very much. You're an amazing hostess. And I want to wish everyone good evening. I'm obviously really looking forward to telling you a little bit about our research, but I'd be remiss if I didn't open my remarks by extending a word of thanks, particularly with Thanksgiving just around the corner. So first off, I'd like to thank each of you for joining us on this webinar. I want to thank you specifically for being active learners, and as was mentioned by our previous speaker, for being advocates and for helping to educate others about ovarian cancer. It's only through understanding the disease that we're likely to make disruptive impact on patient care and survival. Second, I want to thank anyone in the audience who has either donated to OCRFA or fundraised on its behalf. I assure you that I was not asked to address philanthropy during this webinar, but I'm so grateful for the support that you and the OCRFA have provided to our lab that I couldn't resist but comment on the vital role that this organization plays in enabling new projects and nurturing young investigators who are still in the early stages of their careers, as Sarah had mentioned. So today, I'd like to tell you about a tale as old as time, Beauty and the Beast. Now, on the surface, the beauty is our immune system, which usually protects us from harm. And the beast is cancer, no doubt. However, I'd like to scratch slightly below the surface, and we'll see why, hopefully, in the next 10 minutes or so. Now, our lab works in an exciting new field known as cancer immunotherapy. It's been called the fifth pillar of cancer care, succeeding surgery, chemotherapy, radiotherapy, and targeted therapy. It is unique from such traditional approaches in that immune cells can proliferate, leading to amplification of the therapeutic response. Also, unlike passive drugs, immune cells can actively home to tumors and concentrate at the site where they are needed. Moreover, these cells can communicate with one another, generating a coordinated response that is particularly challenging for the tumor to evade. The response is adaptive as the immune system can co-evolve with the evolving tumor. This turns out to be critical. But perhaps the most important facet of the response is that it has memory, meaning that all of these features are preserved over time. By way of simple analogy, compare the temporary pain relief that you get after taking a pill such as aspirin with the lifelong protection against polio that you receive when you stimulate your immune system with a vaccine. Now, in the past decade or so, we have learned that the single greatest predictor of patient survival is the density, location, and type of immune cells within a tumor. And interestingly, ovarian cancer was actually one of the earliest prototypes of this revelatory finding. It's hard to believe, but the immune cells are perhaps more important than the cancer cells themselves. This is because context matters. The cancer cells are like a seed, and the immune cells are like components of the soil. If you plant a seed with perfect genes in lush soil and add water and sunlight, you're likely to grow a beautiful flower. However, if you take the exact same seed with the exact same perfect genes and plant it in the desert, then you are not likely to grow anything at all. Environment is essential. Now, the first study concluded that more T cells, which are a specific kind of immune cell, inside the tumor led to improved outcomes for ovarian cancer patients. But a paper that was published shortly after revealed that not all T cells are the same. In fact, there are CD8 T cells that kill cancer cells, and there are other T cells called regulatory T cells that prevent our immune systems from going into overdrive. This is bad in the context of cancer, but it is critical to prevent us from killing ourselves from autoimmune diseases such as MS, diabetes, and lupus every time we catch the flu. We need to have an off switch under normal circumstances. The next piece of the puzzle indicated that it was the ratio of the cancer-killing CD8 T cells to regulatory T cells that is associated with a favorable prognosis. In particular, the CD8 T cells had to be in the tumor bed itself and not simply on the margin. In the bottom right, you can see different immune cells that are stained in brown and different locations within the tumor. These data show why it's so important to answer basic questions about how the immune system and tumor interact. So if CD8 T cells are so important to patient outcomes and cancer immunotherapy acting on CD8 T cells, boosting their function, why aren't more ovarian cancer patients responding favorably to immunotherapy? I'm glad you asked. It's a great question, and one that we are actively studying. It turns out that there are many more immune cells in the tumor than simply T cells. We'd like to get to know our characters a little better so that we can solve this mystery. And to quote William Shakespeare, all the world's a stage, and all the men and women merely players. They have their exits and their entrances, and one man or woman in his or her time plays many parts. And I think this is really important, the concept of playing more than one part. So while many scientists are actively engaged in understanding how different immunotherapies benefit mouse models of cancer, we decided that we would focus on the most translationally relevant context, the period of time after administration of standard of care chemotherapy. Nearly all ovarian cancer patients receive platinum and taxanes as part of their course of treatment. So we thought that it would make good sense to consider how these medicines alter the composition and function of immune cells around the tumor, as it is known that certain immune cells are more sensitive than others to chemotherapy. We studied which cells, what they are doing, how long this takes to happen, where in the body these cells are, why they are acting the way that they are, and how we can make use of this information to improve mouse and ultimately patient survival. Now to return to our theme, Beauty and the Beast. I think that we can all agree that Belle is one of the good guys or gals. Let's call her a CD8 T cell. Similarly, Gaston is a bad guy. Let's call him a regulatory T cell. But who is the beast? Is he a foe or a friend? It turns out that there is a type of immune cell called a macrophage, and it can be helpful or hurtful. We are currently evaluating drugs that help the macrophages to be more helpful. And returning to the concept of environment mentioned previously, we find that other characters nearby can greatly influence our quote-unquote main characters. We thus try to course correct the patient's immune system to do what it naturally does, which is eradicate tumors. And we're hopeful that the information that we gather will be enabling in this regard. I'd like to thank the members of my wonderful lab who do the research and who are really fun to work with on a daily basis. And also, I'd like to thank again you and the OCRFA. I imagine we're all looking toward the same goal, which is to live happily ever after. So thank you very much.
Thank you, Michael. That was great. Okay, so we have some time for a few questions. Let me see. I've been trying to keep track of them as they've come in. All right, where to start? So I'm going to unmute all of our speakers so they can all talk and weigh in as needed. Because the Cancer Genome Atlas came up, and we got a little bit of a tease, but not a ton of information, I'm wondering if you could, if you guys could talk a little bit about the Cancer Genome Atlas, what it is. We got some sense of, you know, the type of information that the project led to. If you guys could just talk a little bit about that.
Well, I guess I will have to take that question because I'm the only participant of TCGA here.
That'd be great.
So, TCGA is an NCI-led national effort to fully characterize about 500 tumors for each of the major tumor types that you can think of. And and some rare tumor types are included as well, but the numbers there are usually smaller. And the aim is to generate a full molecular atlas so hence the name, the Cancer Genome Atlas, for each of these 500 tumors. And by comprehensive molecular atlas, the goal is to generate genetic sequence, copy number alteration, DNA methylation or epigenetic alteration maps, protein, RNA expression, and protein expression, all for the same tumor, and we will also have matching clinical information for the same patient so that we can do a lot of correlative studies between the molecular alterations and the clinical features, including clinical outcome. And that has been actually quite productive and fruitful. The only limitation is for ovarian cancer research is that all ovarian tumors samples included for TCGA were of the serous histology subtype. And I don't know how well, how familiar the audience is with ovarian cancer histologies, but serous ovarian cancer is the most deadly and most common ones, but there are also other subtypes that actually also need to be researched, but not included in TCGA yet. Is that sufficient answer, Sarah? Or?
Yeah, no, I think that's great. Thank you. Okay, let's see. Next question. Here's one. How is a patient tested for epigenetic alterations? Is that possible?
Well, there are, sorry, I'm jumping on this again because that's
okay.
For me. So, unfortunately for BRCA1 promoter methylation, there isn't any yet because it was not, usually it takes 10 years or to 20 years for discoveries in the lab to be translated to the bedside. But a collaborator of mine is actually looking to do that for BRCA1 and another closely related gene called RED51C. Both are in the same pathway, and both have promoter hypermethylation in ovarian cancer. And so that's still under development. And there are epigenetic testing available for other genes. For example, MGMT hypermethylation in brain cancer is predictive of a response to alkylating therapy. So that has been, it's now being used in the clinical setting now. So hopefully the BRCA test that we're working on will be available soon.
Okay, great. Next question. How is NextGen sequencing different than immunohistochemistry? Anyone?
I think Ron's on mute right now. I'm happy to answer it, but I'm sure he could do a better job than I.
So I took my mute off, but that's a great question. I think that oftentimes immunohistochemistry is used quite readily to detect some of the same molecular features that could be found with next-gen sequencing. So there's definitely tests using immunohistochemistry to look for example, maybe a feature that may be something like a P53 related type signature or something like that. Those same features can be detected by next-gen sequencing. Immunohistochemistry is sometimes a little bit easier to use in the clinic because oftentimes if they've done a biopsy or something on the patient, they have tissue. This can be done in most hospitals throughout the country. Not all hospitals have next-gen sequencing technology. Although this technology is growing, and I think in the future we'll see more of it being used in the clinic.
Yeah, and I think
hopefully that answered your question.
A second point to complement Ron's response is that the next-gen sequencing works on DNA and can be RNA as well, while the immunohistochemistry is typically looking at the protein level. So you get different layers of analyses, which can be very complementary, as Ron mentioned.
Yes.
Yeah, that's a good point.
Yes, I second what Michael is saying, and for some of the alterations can only be detected with the sequencing, on the DNA or RNA level, but because the protein is not quantitatively or qualitatively changed enough for the IHC to have it picked up, so you have to actually use the sequencing to detect it. For example, BRCA1 or BRCA2 mutation, people are still using sequencing, but more targeted sequencing. So you just look at 1 or 2 genes instead of the high throughput next generation sequencing.
Okay, thank you guys. We have a few questions that are all kind of similar and all focused around this. The one question that came in, foods and environmental toxins can cause many epigenetic changes. Are any foods being studied to promote tumor suppressor genes and silence oncogenes? And we have a few questions that sort of focus on food, exercise. Do we know if these things are useful?
I suppose since epigenetics is mentioned, it's a question for me again. But I don't want to keep on jumping to this. As far as food goes, the scientific evidence is actually not very clear on that, and that goes into the more epidemiological aspects of epigenetic studies. And currently, those studies have problems, in my opinion, because they're often looking at the wrong tissues, because different tissues have different DNA methylation, and they're confounded by a lot of issues because the most easily accessible material is the blood, and people look at whole blood, but the different components have different methylation status, and if you have a shift in, for example, white blood cell count, then the readout of those epigenetics will be shifted, and any change in blood cell composition can actually confound the methylation measurement that people get out of these tissue samples. So I don't really see any convincing evidence in terms of food for turning on or off genes. It's certainly not in a targeted way for important genes. But if we are talking about exercise, it is well known that, for example, obesity is a risk factor for ovarian cancer. So through exercise, if the BMI gets reduced, then yes, the ovarian cancer risk can be reduced that way. Michael and Ron,
Great.
do you have any?
Yeah, I was going to add, just a lot of the population-based studies are kind of mixed, and there's not a good smoking gun. But I do think healthy lifestyle habits are good, and I think any clinician is going to tell you to stay healthy, either through exercise, through eating healthy, and I think that's always good advice to have. I think healthy lifestyles are good at cancer prevention. I think we know that in general.
I completely agree with Ron, and in fact, there's even some data emerging to mechanistically explain that, in that stress, for example, and certain, let's call them environmental factors, can be poisons to the immune system, essentially. And so if you have a healthy lifestyle that involves good nutrition, exercise, you're actually boosting your immune system.
Thank you. And Michael, there was another question which I was going to ask next, which I think you basically just answered, which is how can we boost our immune system to remain in remission? And while obviously there are no guarantees about remaining in remission, it sounds like, is that what you would recommend in terms of boosting immune system?
Yeah, absolutely. So, for example, stress is known to be very harmful to the immune system. And so people who meditate or do yoga or exercise, in some cases prayer, by figuring out what works for them in order to alleviate their stress, that's actually helping their immune system continue to perform immunosurveillance.
Great. This is sort of an interesting and kind of, I just thought it was an interesting question. So without BRCA1 or 2 mutations, some doctors label ovarian cancer as just quote-unquote bad luck. How can this be bad luck? And I've heard that as well, sort of chalking up to, well, most cancers that people end up getting are just kind of random. Do you guys have any commentary on that?
Yeah, that's a very interesting study that came out of Johns Hopkins between a very renowned cancer biologist and a mathematician. And I think it's very provocative, but it's hard to conclude one way or the other at this point. It also makes one consider, what is a tumor-initiating cell versus a different type of cancer cell. So I would not feel comfortable to make a definitive assessment on that. I think it's interesting in that it frames a question that we can hopefully delve more deeply into. But I think if we all understood, if we thought it was just luck, then we wouldn't be able to really find all the answers that we're finding in basic biology. But conversely, there are factors we don't yet understand.
Right. Thank you. Let's see. I'd be interested to hear if you guys have any commentary on this. There was a question that came in here about if a person has had their tumor sequenced, these days, given how quickly the science is changing, if someone were to have their tumor sequenced and were to get back information about their tumor, how clinically actionable is the information that is available these days that patients can get if they were to, say, have their tumor sequenced? How useful do you think it is today? I think certainly it seems like in the future there's a lot of potential there. But beyond knowing BRCA mutation status, is there anything essential that people should be looking for that might actually help them with treatment decisions for a person who's undergoing treatment now?
Well, I don't think we should underplay BRCA. In fact, I would argue that's one of the poster children for targeted therapy or precision medicine.
Sure.
So for women with gynecological malignancies, I think there's tremendous value in having genomic testing. But certainly there are other alterations or genetic lesions that are in clinical development under investigation. One that comes to the foremind is PI3K alteration, PI3 kinase. So that's one to look out for, but that's really for a clinician, and particularly your clinician, to answer.
Right.
Yep. Sarah, you actually raised a very good point. Sorry, I did not mean to jump in if you're trying to conclude.
Oh no, I'm all done.
No, please go ahead.
I heard you talk. So basically, you raised a very good point because a lot of the mutations are still considered not actionable right now.
Right.
but discoveries are being made, and you know, as you said, in the future we'll actually know more and more about what these mutations do. And for example, ARID1A mutation that Ron talked about, it was only discovered quite recently, and those are in the non-serous ovarian cancer. So those are in the endometriosis-related subtypes. And I think in 2015, MD Anderson had a study that showed that if you have ARID1A mutation, the tumors respond better to PARP inhibitors. So a lot of the trials right now are actually dealing with, so they do not stratify the patients by which site of occurrence of the tumor is, but which mutations they have. So there are so many trials going on right now that we will see a burst. And that's my hope, that we'll see a burst of information in this part, that soon we will know how to actually better inform treatment with all these mutation information. That
yeah, that's an excellent point, because I think we, in the news, hear a lot about BRCA, but BRCA is really one particular mutation in an important pathway, which is involved in DNA repair. And there are many other genes that encode proteins in that pathway and related pathways, and one was just mentioned by the last speaker. So I think we don't yet know how all the pieces fit together, and that's why some of those genetic alterations that can be found with current technologies may be drug actionable, you know, tomorrow or the next day, even though we don't know it today.
Right. Okay, terrific. I think maybe we will wrap up now. So thank you, a big thank you to all of our speakers for these terrific presentations, really fascinating and also exciting to get a glimpse at where the science is headed. And I want to thank everybody on the phone who called in today. Please take a minute to fill out the survey that will appear on your screen, or should appear on your screen at the end of the event tonight. Your feedback is always helpful to us, especially as we put together future webinars. And again, this webinar has been recorded, so you'll be able to listen to the audio and see the slides again. It'll be posted on our website. And for more information about OCRFA, you can always visit our website, www.ocrfa.org. So thank you, everyone, and have a great evening, and happy Thanksgiving. Bye.